So we were talking about the movement of blood through the four-chambered heart, and we have, with the movement of blood, the contraction and then subsequent relaxation of the major chambers in the heart, and that is what is referred to as the cardiac cycle. So when we look at the cardiac cycle, we can talk about the systole part of it. So systole equals contraction. And then diastole, which is kind of a weird way to say it, but I'll talk about diastolic in a minute, is relaxation. So systole, when we think about systole and diastole, oftentimes we think about blood pressure. So we could talk about systolic versus diastolic blood pressure. And this is actually directly related to the ventricles. So this relates to what is happening in the ventricles. So if we look at blood pressure could be like 140 millimeters of mercury over 90 millimeters of mercury. And that is like 140 over 90. That's kind of the high end break point. Like if it's over 140 or over 90, that tends to mean that you have hypertension or high blood pressure. So this is measured, we're going to talk more about this, but this is measured generally in your brachial artery. And so systolic pressure, this would be your systolic pressure, this is the large number. That is when the ventricles are contracting. And so that means that there's lots of blood being pumped out through the brachial artery. And then this is your diastolic pressure, and that is when the ventricles are relaxing. So that would be the low pressure. And so it's important to realize, however, that we can also have systole and diastole, not only of the ventricles, but also we have to have it in the atria as well. So if we look at in your book, there's a little diagram that shows this. When blood comes in to the atria, this would mean that the atria would have to be relaxed, and then the ventricles would also have to be relaxed. So the blood comes in, and then the atria would then contract to actually force blood into the ventricles. And then when the ventricles contract, we want the atria to be relaxed, okay? So there's a cycle of alternating relaxation and contraction of the different blood vessels. So atrial systole just forces the blood down into the ventricles, and then ventricular systole would force blood up and out of the heart. Okay, so this occurs, I'll put, just occurs in the atria as well.
That happens like every second?
Yeah, so every time your heart beats, yeah. So if you're speeding 75 beats per minute, that would be even a little bit more than every second. So the atria relax, the atria contract, the ventricles relax, the ventricles contract, and it's a cycle, okay? So you just need to know systole means contraction, and diastole means relaxation of the chambers in the heart. Okay, and it relates to blood pressure when we're talking about the ventricles specifically. Okay, so when we look at this cardiac cycle, oops, we already talked about that. We can also talk about how the heart rate is regulated. And generally, we measure heart rate in beats per minute. So BPM is beats per minute. Okay. So we have two mechanisms. One is the intrinsic regulation. So when you think intrinsic, think inside, internal regulation. And so this has to do with the pacemaker of the heart. So this has to do with special tissue in the heart, and this tissue is called nodal tissue. So this special nodal tissue in the heart can initiate contraction of the heart. So I'll just say can initiate. And so it actually has some characteristics of nervous tissue. And when we talk about contraction, there's a word which we haven't talked about because we haven't gotten to the nervous system yet, but it's called depolarization. So sometimes in your book and elsewhere, you'll see the word depolarization, and that is what is happening when the heart and the muscle contracts. It depolarizes and then it contracts. And we're going to talk a lot about depolarization when we get to the nervous system, because that is also what happens inside the neurons when they're sending an action potential or an impulse. They become depolarized. Okay, so I just want you to realize that contraction and depolarization are sometimes used interchangeably. Okay, so in your heart you have what is called the sinoatrial node. This can be abbreviated as SA node. This is in the right atrium. This is your pacemaker. So this is like the place that initiates the contraction of the heart. Now the heart muscle cells are actually connected to one another. And so when one heart muscle cell contracts, it will cause another cell to contract. And so you can get a wave of contraction moving through the heart. Okay, so the SA node is your pacemaker. Now we also have some nodal tissue, which is called the atrioventricular node. Yes?
The SA node, is that the first electrical shock mark gets? Because it gets like 3 or 4 of them.
Yes. Yes. Yeah. Yep.
So
that's the first one. Yep. So then it goes to the atrioventricular node. So this is called the AV node. And this is still kind of near the right atrium, but it's also near the ventricle. So remember we talked about atrioventricular AV valves, right? And so atrioventricular node. So what the AV node does is it delays the signal from the SA node. Really short, like it's like 0.1 seconds, right? So it's not very long, but what that does is it allows the atria to relax before the ventricles contract. So it allows atrial diastole, right, before ventricular systole. Okay, so it allows the atria to relax before the ventricles contract. So this is important because we actually want it delayed, and then we actually also want the signal to not start at the top of the ventricles, but we need the signal to start at the bottom of the ventricles. So we also have some nervous tissue / muscle tissue that is called the AV bundle. So I'm just writing that is also atrioventricular, right? So AV bundle, the atrioventricular bundle. This transports the signal, right, the depolarization, to the bottom of the ventricles. And in the heart tissue itself, we have what are called Purkinje fibers. And this is named after a person. That's a J, Purkinje fibers. And the Purkinje fibers are what cause the ventricles to contract from the bottom up. Okay. And this is really important in a 4-chambered heart because you wouldn't want them contracting from the top down, because then the blood would be forced against the wall of the heart, and eventually might even erode the heart wall and cause a hole to form in the heart, right? So we want the heart to pump from the bottom up so blood will flow in a laminar fluid fashion up and out and into the blood vessels, okay? So this is all intrinsic because actually you can do these really crazy experiments on reptilian hearts where you can actually remove, so when I was a graduate student I had to do this, which I didn't want to, but the professor made me. But you can actually remove a heart from a turtle by taking and drilling a hole in the carapace and pulling the heart out, and it will just beat on its own. And it's super hard, it's super hardy. And so in order to actually get the heart from stop beating, you know, outside the body of the turtle, you actually almost have to mutilate it. So you have to cut it in tiny little pieces and then throw it away, because you don't want the janitor to come in and see something beating in the bottom of the garbage can, right? So. But anyway, so it's really interesting that the heart can beat on its own due to its intrinsic conduction system, okay? So this is the handout that I gave you. So this is your SA node. This is your AV node. This is your AV bundle. So it goes down between the ventricles. There's a wall, a septum, that takes it to the bottom of the heart. And so then these little fibers right here that innervate the heart are called the Purkinje fibers. So that is all intrinsic conduction. So we also have extrinsic conduction. You could write this probably back in your notes so you know. So this is outside the heart, extrinsic. This would be a really good essay question for the quiz on Wednesday, right? What's the difference between intrinsic and extrinsic, right? So extrinsic conduction has to do with how the nervous system can innervate the heart and change its heart rate, okay? So this would be nervous system, and we could call it innervation. So the autonomic nervous system has two divisions. One is called the sympathetic division. So if we have sympathetic innervation, this is the fight or flight, and so this is going to increase your heart rate. So it will speed it up. When you're exercising, your sympathetic nervous system takes over, and it causes an increase in your heart rate in order to deliver oxygen out to your muscles. And then the parasympathetic is your relaxation, digestion. So this is going to decrease your heart rate, okay? So these are just parts of your autonomic nervous system. So I'll write that down. So these are components or parts of autonomic nervous system. And so, for example, we have nerves that extend down from our brain and then go and connect to the SA node, and they can send a signal to the SA node to speed the heart rate up or decrease heart rate. Okay, so when we talk about innervation, that means that there's neurons that actually synapse, they actually make connections with the nodal tissue and cause contraction to occur. Okay. We can also have hormones. Right? So the nervous system is one form of extrinsic. I'll put regulation, sorry. Let's not do conduction. That was kind of confusing. I meant to say regulation. Sorry, regulation. So we have hormones. So primarily, when we think about the heart, it responds to hormones that are produced by the adrenal glands. And so adrenaline. right? And this is also called epinephrine. Those are produced by the adrenal glands, adrenaline. So this would speed up your heart rate. So you could do experiments on turtle hearts where you can actually just douse them with more epinephrine and the heart rate will increase. But in general, it's the blood that carries the hormones to the heart, and then the hormones bind to target cells, and then they would cause the heart rate to speed up. So this tends to be long-term, right? And this would be more of a short-term regulation. Hormones tends to be like long-term increases in heart rate. So over a period of hours, for example, adrenaline can cause your heart rate to stay elevated. Okay, so when we look at the excitation of the heart, we can look at how the electrical impulses move through the heart. And when we do that, we can measure it using an electrocardiogram. So this is abbreviated as ECG. That's an electrocardiogram. Now, sometimes they call this not ECG, but they call it an EKG, which is the same thing. So ECGs and EKGs are the same thing. But why do you think they have a K instead of a C? Anybody know? German. Okay, so in the German language, cardio is with a K. And so if you're talking about in the German, if you use EKG, you're actually using the German terminology. Okay, so the electrocardiogram just measures the electrical signal across the heart, and it looks something like this. So it starts out, and then you get this depolarization right here. So you get a little wave, and then there's a picture of this in your handout on the back too. And then it kind of goes down, and then it goes up, and then it comes back down, and then it goes steady, and then you get a little blip at the end. So this is kind of what an EKG would look like. So it's talking about the change in voltage, in potential, membrane potential across the surface of the heart, okay? So this is what we call the P wave. This right here is called the QRS complex. And this right here is called the T wave. And then you would repeat it again. So when you're hooked up to a machine and it's going blip, blip, you know, it's measuring your heart rate, you can actually measure your heart rate because this is, every time this happens, your heart will contract, right? But you're also making sure that the electricity is moving through the heart in the proper direction, okay? So P waves, this is atrial systole, right? So that is where the atria contract. So this would be contraction right here. So that's the P wave. The QRS complex is ventricular systole. And at the same time the ventricles are contracting, the atria are also relaxing, and so it's actually kind of hidden underneath QRS. You can't see when the atria would relax because it's the exact same time the ventricles are contracting. And then lastly, we have the T wave, and this would be when the ventricles relax. So this would be ventricular diastole. So in the handout that I gave you, you can see what is happening as the electricity moves through the heart. Okay. So P, right, the SA node sends out its signal, and that depolarization causes contraction. And then QRS would be when the, actually it's showing it right here. QRS is actually when the ventricles are contracting right here, right? And then the T is when the ventricles start to relax. Okay, so remember, depolarization just means contraction, and repolarization would be relaxation. Okay, so they're showing, as it goes through, they're showing where it is. So QRS, this is ventricular, it's just finishing its contraction, right? And then here it is, it's starting to relax. Okay, so sometimes you get really weird things happening, and you can get imbalances or defects in the heart, and one is what is called atrial fibrillation. Okay, and so what this is the atria are not contracting properly, so it kind of looks like this. So you get these peak, kind of weird, funky contractions, and then you get the QRS, and then you get, oops, I didn't do my QRS right. Then you get a T wave, right? So you get abnormal P waves. Now, this is actually one of the most common problems with the electrical conduction in the heart, and it's commonly called AFib. So you hear older people all the time talking about their AFib and the drugs they take to control it, right? And so you can actually, when a person has this, you can listen to their heart, and it sounds like this. It goes, and it's kind of, sometimes they say my heart is palpitating, right? And it can also sometimes go along with fast heart rate or tachycardia, okay? So the reason for this particular problem is that you have extra nodal tissue. So extra nodal tissue causes abnormal contraction of the atria. Right? So there is actually a really effective cure. It's actually a cure for AFib, and it is called ablation. And ablation is where they go in and they freeze or burn that extra nodal tissue. So they freeze or burn. Right. So they can actually go into the heart up through a vein. So they take a little catheter up through the vein, little piece of metal, and then they go into the right atrium, and then they find the part that is firing inappropriately, and they will burn it. And then that will help to prevent AFib from happening. Right? And this can be kind of a problem specifically for people who are athletes. So if you're an athlete, like a long distance runner or a bicyclist or somebody who does like long aerobic sports, sometimes this is a common side effect of that. This is that when you put your heart under stress and your heart kind of gets big, specifically when the atria start to get big, because they're receiving more blood and you have more of a cardiac output, then you get this extra nodal tissue forming, okay? This is not really life-threatening, but it can be really bad. Like, it can make you feel really bad. Like, it's really, if you can imagine, it's really hard to sleep when your heart is like going, crazy and doing crazy things. And so it can make you feel really bad. And then the drugs are not very nice in the fact that they have some bad side effects. But ventricular fibrillation is much worse. Right? So this looks like this, right? It's just like crazy, maybe not like that, but it's crazy, right? And so there is no normal relaxation and contraction of the ventricles, and so this can lead to death, right? And so how do you treat ventricular fibrillation? With right? You shock them. So you use what is called a defibrillator. So you use the paddles, right? And you put them on the body and you shock the person. And what the hope is if you stop the heart and then it restarts on its own, then what is going to happen is that normal sinus rhythm, the normal ECG, will take over, right? And you'll get the heart rate coming back. Okay, so that's the basis for shocking the heart. And there's actually, we now have automatic defibrillators around like campus. Sometimes you see them, right? Which are pretty self-explanatory and are easy to use. A heart attack can cause ventricular fibrillation, as can like electrical shock. So if you get shocked, if you're messing with electricity and you get shocked, sometimes it can cause your heart to go into ventricular fibrillation. Okay, so that's the electricity in the heart, the electrical conduction system, and how it is regulated. So next we're going to start talking about blood vessels. And when we talk about blood vessels, remember that blood vessels are actually organs. So we saw this in our slide in lab last week, in that they are composed of different types of tissue. So when we look at the arteries, these by definition carry blood away from the heart. Okay. They also they're carrying blood at higher blood pressure, and they generally are carrying oxygenated blood. Okay. So blood higher pressure, except for the pulmonary arteries, right? Those are unoxygenated. And because they have higher pressure, they have a thicker layer of smooth muscle. They also have elastic connective tissue. And you can understand this when you think about your pulse. So when we're like measuring a pulse, the elastic connective tissue, the blood causes the vessel to bulge out, right? And then it would contract against. So it kind of keeps the blood vessel from getting too big, right? And then it causes it to kind of bounce back. And so that elastic connective tissue is really important, specifically in the deep arteries, like in the carotid artery, for example. Okay, so this is generally oxygenated. So I'll just put oxygenated blood, except, right, the pulmonary artery. Okay, so the pulmonary artery is carrying blood to the lungs. Okay, we also have veins. And so in the veins, this is carrying blood back to the heart. This is lower pressure, less smooth muscle. So they're thinner walled. Right? The veins have valves. And these specifically help prevent backflow. Help prevent backflow. Because the blood is under such low pressure that sometimes it has a hard time moving up against gravity. So if you think about the blood from your feet can sometimes pool in your feet, and it's hard for the blood to get back up. And so sometimes you have problems with transporting the blood, and you get what are called varicose veins. And so varicose veins are where you get damaged, and most of the time you can see them if they're superficial, right? And it's damaged veins where their valves have been damaged, and the blood will pool, and it will kind of cause them to get big. And so some people have really, and it tends to be a genetic thing, will have really ropey looking legs, and that's because the valves in their veins don't work very well. and the blood kind of causes those veins to expand. And I've actually known people that have gone in and have had a lot of those veins ripped. They call them stripping them. So they actually remove those veins from the legs. And then they also, because they don't have a lot of veins, they also wear compression socks. So sometimes people have to wear socks that have a compressive force to them. And they're kind of weird in that you can actually figure out how many pounds per inch a sock, a compression sock, would force onto your skin, right? And that helps to keep the blood moving up because it keeps the veins partially contracted, right? So these veins normally are associated with skeletal muscle. So as the muscle moves, so as you move, blood will be pumped back towards the heart. So this is why it is really hard when people have jobs where they're supposed to stand in one spot and not move, right? Because eventually you might have damage to your circulatory system because you're not moving enough to keep the blood flowing from the extremities up back towards the heart. And so then that might be a environmental cause for varicose veins and having to wear compression stockings so that you'll be able to get your blood to move back up. Okay. When we look at the capillaries, these are very thin-walled vessels. So they're very thin, narrow vessels. And this is where exchange occurs with the interstitial fluid. So this is the fluid that is found inside the tissues. Interstitial fluid. So each of your cells is bathed in interstitial fluid. Okay. So the blood has to get into the capillaries in order for oxygen to diffuse out, carbon dioxide to diffuse in order for nutrients to be delivered and waste products to be picked up. And so we're going to talk about capillary exchange in a minute. Okay. So those are the capillaries. Okay. So in your book, they show the difference between the arteries and the veins, right? So the veins here you can see have thinner, smooth muscle, and we saw that in the histology too in lab. And then you can see the presence of the valves. So as blood gets pumped here and then flows back, it would actually close that valve. So it's actually a mechanical valve. And so it closes the valve so that the blood will go all the way back down to the feet. So it just kind of helps it move up in a fashion back to the heart. Okay, okay. So if we look at the blood vessels, right, you can see that there's blood vessels that take blood up to the brain. So your carotid arteries, for example, and then the jugular veins that would take blood away from the heart, or excuse me, away from the brain and back towards the heart. And so we have a circulatory system that branches, and you'll notice that the arteries branch as they come out, and then the veins reconverge. So as they come up from the legs, they would reconverge into big blood vessels that would eventually transport the blood into a really large blood vessel, the inferior vena cava, and then back to the heart. Okay, so let's look at blood pressure. So again, blood pressure is measured in millimeters of mercury, right? And we talked about atmospheric pressure. That's the same same unit in atmospheric pressure. And it's actually the pressure exerted on a certain amount of mercury, right, a column of mercury, and how it presses down on that column of mercury. It's kind of interesting. Anyway, so that's the blood pressure. And so when we look at factors affecting, so factors determining, okay, one would be the volume of blood. So that would be the amount of blood. And blood has red blood cells, but it also has water in it, right? So it has plasma. So plasma, blood plasma, is water plus dissolved solutes. One of the things that is in plasma is sodium. Sodium ions, chloride ions, calcium ions, electrolytes. The other things that are in them include glucose, amino acids, proteins, right? All these things that are dissolved in it. So we'll say that there's ions, right? So that would be like sodium, chloride, potassium, calcium, right? There's proteins in solution, like hormones, right? There's also glucose. Okay. The reason I mention this is that sometimes we can have too many solutes, and that will actually affect the volume of the blood. And so an example of this would be if you take in too much sodium. So here's my example. Okay. So let's say you eat too much, you take in too much salt. Your body compensates by retaining water. Right? So your body retains water in order to dilute the sodium and the chloride, right? And so this increases blood volume. So what can that do? That can cause hypertension. So hypertension means that you have high blood pressure, right? And it's generally like anything over 140, over 90 is hypertension. So hypertension is the same thing as high blood pressure. Okay. So that's why your grandpa, when he says, oh, I can't eat salt anymore because of my blood pressure, right? And that's the relationship between that, right? So one thing that you can do, if you can change your diet, right? But what else could you do to decrease the volume of water in your blood? What could you take? Not blood thinners. Nope. Because we're not talking about clotting yet.
Like diuretics.
Diuretics, right? So you can take a diuretic. So let's say, oh, well, of my blood pressure, they put me on diuretics, right? So diuretics, diuresis means to urinate, to get rid of water. So this increases output of urine, right? So this would decrease the volume of blood because you would lose water in your urine, and that decreases the volume. So that would decrease your hypertension. So sometimes when people have high blood pressure, they're on diuretics because of that. Okay. Okay. The second thing that can affect blood volume, so this would be number 2, right, is the diameter of the blood vessels. So when the blood vessels constrict, it's called vasoconstriction. Vasoconstriction means that the smooth muscle in the blood vessels have constriction, been activated, and so this decreases the diameter. And when you decrease the diameter and the volume's the same, you increase the pressure. Right? So it's kind of like you have water flowing through a hose. You put your thumb over the end of it, you decrease the diameter, but the water now comes out at a very high pressure, right? So the opposite of vasoconstriction is vasodilation. So this is where you increase the diameter, but you decrease the pressure. Now, normally what happens is that blood gets shunted away from vessels that are constricted, but sometimes blood really needs to go to a certain place like the heart or the brain, and so it goes through there anyway under high pressure, and sometimes it can cause the blood vessels to rupture, right? So this would lead to vasoconstriction, this would lead to hypertension, right? Vasodilation would decrease blood pressure, okay? Decrease in pressure, okay? So one problem that we have oftentimes with our circulatory vessels, this is an example of how diameter affects, is what is called atherosclerosis. So atherosclerosis is where we get plaque buildup in the walls of blood vessels, specifically arteries. So plaque buildup. And this has to do with cholesterol. So the plaque contains cholesterol in it. And it's kind of interesting because cholesterol cannot be transported in the blood without being bound to a protein. So when we look at cholesterol levels, we sometimes talk about the difference between HDL and LDL. So HDL is high density lipoproteins. Right? And LDL, that's lipoproteins, that doesn't even look like that. Lipoproteins. LDL is just low density lipoproteins. Okay. So these are proteins that transport cholesterol. So does anybody know which one's good and which one's bad? No. HDL is good. So they say, oh, that's good cholesterol. But really they're talking about the lipoproteins that transport the cholesterol. Because cholesterol is cholesterol. There's no good or bad cholesterol. They're looking at how it's being transported. And so these transport cholesterol from the liver to the tissues. Okay. So LDL is bad because what happens is it keeps the cholesterol in the blood and where it can eventually get into your arteries and cause plaque buildup, okay? HDL is good because we take it out of the tissues and we store it in the liver. So it would be stored here, right? So it's taken out of circulation and stored. And so my family has, it's kind of, the doctor thinks just genetic. So genetically, we have too much LDL, which puts us at a risk for atherosclerosis, right? So what can happen with the atherosclerosis is that you can get a couple of things happening. You could get an embolism. Actually, let's just talk about strokes. So in a stroke, you can get a blocked blood vessel to the brain. Or you can also have the blood vessel rupture. And it causes the same effect that causes the death of some brain tissue. And so this causes death of nervous tissue. So it's important for people to recognize the signs of strokes, right? So whenever people have like a droopy, like half their face droops and the other half doesn't, or sometimes they have difficulty speaking because their tongue doesn't work properly. And sometimes they also, like if they lift both arms and one arm, they can't lift it. It's weakness in that arm. That is also a sign of a stroke. So strokes really only happen on one side because it's generally only on one side of the brain that the blood vessel has been damaged. Okay. So they talk about stroke attacks being able to detect the signs versus like heart attacks, right? So in heart attacks, we get death of the cardiac muscle. And so this is a blocked coronary artery. Right? And so we can get death of muscle tissue. And so the muscle can be weakened to the point where it is harder for it to pump, and then you can get heart failure, or it could just cause death. Right? So if the heart just stops beating because it is deprived of oxygen, then it can lead to heart attack. Okay. So I'm going to show you just a short little video that shows a person that has blocked coronary artery, and when they do a bypass surgery, they bypass the blockages, but they can also put in stents in the blood vessel.
Our lives depend on the health of our coronary arteries. Yet 16 million Americans suffer from coronary artery disease. Georgia Chan Downard is about to discover that she is one of them. She never suspected that, like so many others, she was at risk, but her doctor did.
My doctor thought it was about time I had a stress test. I could tell as he was looking at the graph that there might be something. Maybe it was instinctive on my part. Maybe I was just nervous. And he turned to me and he said, there are some irregularities here. I didn't really understand until I said to him, well, exactly what does this mean? And he said, I think maybe you have a blocked artery.
Heart disease was far from Georgia's mind. She had no obvious symptoms, no chest pain, no shortness of breath. But Georgia did have high cholesterol and a family history of heart disease, which worried her doctor. He recommended a closer look. A non-routine procedure using a highly sophisticated CAT scan that can picture the beating heart in 3 dimensions.
This is what your heart looks like in your chest. It's not like an artist's rendition. It's really actually what it looks like. Here is the left coronary artery that comes off of here. This artery looks very large and healthy up here. But then suddenly there's a big change from this size artery down to this size artery. And then it kind of opens up again. This right here is the area where it looks like significant blockage.
Because her artery was severely blocked and appeared vulnerable to imminent heart attack, her doctors decided to insert a stent, a tiny mesh tube to open the narrowed artery. Stenting is a routine procedure, but very recent studies suggest that some patients may do as well with intensive treatment with drugs. Georgia's hidden heart problem is due to atherosclerosis, deposits of plaque in the artery wall. In healthy coronary arteries, the blood flows without obstruction. In diseased coronary arteries, a fatty substance called called plaque forms in the artery walls, and as the disease advances, may bulge out into the path of the onrushing blood, blocking the oxygen and nutrients the heart desperately needs, and from time to time, causing symptoms like chest pains and shortness of breath. But heart attacks result more often from plaque that does not protrude into the artery itself, but remains hidden in the artery wall.
One of the scariest things about coronary artery disease is that it can remain silent for so long. You can have plaque buildup in the coronary that isn't causing you any problem, doesn't cause you any symptoms, doesn't even cause a positive stress test, but suddenly ruptures. It ruptures, it causes a blood clot to form, the rest of the coronary artery, the rest of the pipe suddenly gets blocked, and a heart attack occurs. And that's really the most common cause of heart attack. This is what kills people, often in the prime of their lives.
All plaque is dangerous, but some plaques are unstable, on the verge of rupture. Scientists are today trying to determine how to identify the unstable plaque, while doctors try and help patients minimize their risks.
The plan right now is to manage the coronary disease in the arteries. Your blockage, if it was up further, up at the very origin of that artery, that's a very dangerous obstruction. And if it closes, generally causes a very extensive amount of damage to the heart. Your blockage was in the mid portion, it would still cause a pretty good amount of damage that would certainly alter your life, but it's not exactly as lethal as the more proximal regions.
I have a small gym in my building. Do you suggest I, go down and exercise?
I would not. I would wait.
No?
I would do
Wait
all of your
until
normal
after the stress
activities. I just wouldn't be breaking a sweat. I wouldn't be getting on the treadmill at this point, only because there's some evidence that you can jeopardize the stent opening.
Okay.
So if you do it too early.
Okay.
So what we're going to do next is.
What a pity. Sorry.
Well,
I'm
you'll
not a
get
big
there.
fan of exercise.
You'll get there.
I know.
And by the time you finish cardiac rehab, you'll love exercise so much you won't
Really?
be able to wait.
Okay. Any dietary restrictions other than the obvious?
The obvious. It's really fat, cholesterol, red meats, egg yolks, cheeses, whole milk, switch to skim.
Okay. So that last suggestion actually has been rescinded about cholesterol, and that you don't really need to watch your dietary cholesterol because it doesn't seem to influence the cholesterol levels in your blood directly. It has to do more with the lipoproteins that transport the cholesterol. Okay, so we're going to take a break, and what I'd like you to do is we're going to go down to the lab, and you might want to bring a piece of paper so that you can make notes about your parasitic fly experiments. One of the things about the parasitic fly experiment that you want to note is the life cycle. And so remember that these go from egg to larvae, which feed, and then they go to pupa. And so we're going to see whether or not our larvae that we saw last week have pupated, and the pupa are going to be maybe a darker color, and they're going to have an exoskeleton over them. And remember, that's when they go from like the maggot-like creature to actually the wasp, right, with the wings. And so that's a big step in metamorphosis. So we'll go downstairs.
was like, oh, well, that didn't work. Man, I've been watching like hella YouTube videos on the beginning, the first half of this class. Like the final day.
And then just
And you know, the progression. I think
that's good. Yeah. Like 2 months.
what parts make it get better for me? Yeah.
Yes.
Yeah.
And then it's more.
That 211 times is full.
Yeah. So basically, create a virus and try and kill the whole world without getting a cure. It is amazing. I don't. Like, like to get them all in life, you have to feed them before. But there's like bacterial, virus, parasites, and then you can do like biochemical and zombie. And so, like, you start off like how you want to spread, like water, and then you bring all the symptoms of it. And then they start working on the cure. So they'd be like, oh, shoot, we should have all works. So it's
Okay. So when you look at blood pressure in the arteries, it varies with the contraction and the relaxation of the ventricles. And so that is why you get the pulse. And so you can see on this diagram, you can see that the blood pressure kind of increases. That would be systolic pressure, and diastolic pressure would be where the ventricles relax. And then you'll notice that when you get to the capillaries, the blood pressure and velocity slow down dramatically. And that is because the capillaries are very narrow and they're very thin, and so there's lots of resistance to flow. So we lose the pressure at the capillaries, and we also lose some of the fluid that is in the blood has to go out into the tissues. And so we're going to next talk about capillary exchange. Okay. So if I drew my capillary like this, blood coming in from the arteries would come in via the arterial end. So the arterial is just a really small artery. It's branched and branched and branched until it's come to the capillary. And then at the other end of my capillary, I would have what is called the venule. And for some reason, the way they spell this, they leave the I. I think I would have left the I in there, in vein. But that's the correct spelling. That's the venule. So that's the other end of the capillary. So pressure coming in, we have high hydrostatic pressure. And so what happens at the end of the arterial is that stuff gets forced out. So for example, water at this end of the capillary, water leaves, right? Oxygen leaves, and nutrients leave. Okay, so we have exchange with the fluid that is surrounding the capillaries and also surrounding the tissues. Now, at the venule end, we need to take up some of the water, and we also need to take up the waste products and carbon dioxide. So we have kind of the opposite thing going on at the other end. So water gets sucked back in, CO2 moves back in, and then we have waste. So for example, we'll talk about the nitrogenous waste like urea is picked up. Okay. Now the reason why these substances move at the venial end has to do with osmotic pressure. So we have, at this end, we have low hydrostatic. So I'll say we have low hydrostatic pressure, but we have high osmotic pressure. And hopefully you remember something about osmotic pressure. If you took biology 211, you know that it has to do with solutes that are in solution, right? So one thing that typically does not leave the capillaries is a blood protein called albumin. So this is my albumin. So it cannot leave. This is a protein in my capillary. And it does not leave, so it doesn't go out. It cannot get across the membrane. It's too big. But what it does is it creates a solute concentration. So once water leaves, albumin is going to be at a high concentration at this end, and water is going to be sucked back into the capillary. So that is osmotic pressure. So osmotic pressure has to do with that you have a high solute concentration and stuff gets picked back up at the venial end of the capillary. Okay, so stuff tends to move, including water. Okay, yes?
Where are the capillaries?
The capillaries are everywhere that you have capillary beds in your organs, you have capillary beds in your skin.
So are they like connected to the ends of arteries and veins? Or
Yes. So our red blood cells never leave the vessels unless the vessels get damaged, right? And so they always stay within vessels. And so if you look at our circulatory system, the arteries branch and branch and branch into smaller and smaller, and then they end in capillary beds. And then the capillary beds are connected to the venules, and then the venules get bigger, 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 bigger.
Okay.
Right?
So it's kind of like in the lungs, the areola? Or the
Yes. So the alveoli would be surrounded by capillaries. Okay. Yep. But even our skin is full of capillary beds, and each organ has its own capillary bed supply, right? So all of our cells essentially have to be supplied with capillaries. So that's a good question. Okay. So high osmotic pressure sucks, due to the presence of albumin, sucks it back up. Okay. If it doesn't get sucked back up, then you get swelling, right? So surrounding the blood vessels, so surrounding capillaries, are lymphatic vessels. And these lymphatic vessels pick up excess fluid that is lost from the capillary. Right? If they can't pick up all the excess fluid that's lost from the capillary, then we get tissue swelling. So too much fluid in the tissue. Oh, so too much fluid in the tissue causes edema, and this is swelling. Okay. And so edema is actually a symptom of diabetes. There's lots of symptoms of diabetes, but sometimes the water concentration in the capillaries doesn't quite work right, and so you get people that have like maybe one leg will be really extremely swollen, and that is because of the fluid that's leaving the capillaries is not getting picked back up correctly, and then it tends to cause pressure and it can cause nerve damage, and it can be just a really bad thing to have swelling the appendages. Okay. Okay, so a picture of this would look something like this. Okay, so here's my artery. It branches into a smaller arteriole, and this is my capillary bed. So these are my small capillaries. They branched. It's high resistance to water moving through there, to the blood moving through there. So it slows down a little bit. Red blood cells stay in there, but we get the movement of substances out, and then there would be lymphatic vessels that would pick up the excess fluid. Okay? They would be associated with capillaries. Okay. So this actually shows the lymphatic. For some reason, the lymphatic system is always written in green. We talked about the lymphatic system when we were talking about the digestive system, because remember, each villi of the small intestine has a lacteal. That would be lymphatic, right? Our lymph nodes are also lymphatic. So they would pick up this excess fluid, and then it would travel through the lymph nodes and then eventually get dumped back into circulation up near the neck. So that's another function of the lymphatic system. We talked about atherosclerosis. Okay, so we're going to actually move on to the next chapter, which is osmoregulation. So we'll start this today and then we'll finish it on Wednesday. So this actually also relates to osmotic pressure. Osmo means solute concentrations. So function of the osmoregulation would be to regulate, or the regulation of solute concentrations in the body, and also the ability to remove nitrogenous waste. So the removal of nitrogenous waste. So what would be our main organ that functions in osmoregulation? What is the organ in our body that is our main osmoregulator? Kidneys, excellent. Okay, so this is the kidneys. This is also sometimes referred to as the excretory system, because we excrete nitrogenous waste, right? So it's part of the excretory system. So if we look at some organisms are what are referred to as osmoconformers. And this means that they are the same solute, they have the same solute concentration as their environment. So same solute concentration as the environment. Jellyfish, right, are osmoconformers, right? So they live in marine environments, salt water. It has a high solute concentration. The inside of their tissues is the same solute concentration, right? So they're not losing water or gaining water from their environment. They conform to the solute concentration in the salt water, okay? So this is good for them because they don't have to use energy. We actually have to use energy to maintain, because we are osmoregulators. So we are osmoregulators. And this means we use energy, right, to maintain internal environment. So example would be the planaria. So planaria that we looked at in lab were actually freshwater organisms. And so they actually would gain water from their environment, and they have to get rid of the excess water so that they can maintain their internal solute concentrations. So planaria, for example, are freshwater, right? They can be marine too, and then they have the opposite problem, but they would gain water via osmosis. So what that means is water just moves directly across the surface of their body, right? So this is a passive process. It's a physical law, kind of. It's just like gravity. It's a force, right? So water is just going to go into their body because they have solute concentrations that are higher inside their body, right? So then they must use energy to get rid of the excess water. Okay. And so if we look at the planaria, they kind of have an interesting excretory system in that their tubules, oh, I don't have a picture of that. I gotta have a picture of that, I'm sure I do. Oh, I forgot to put that on I'm sure I had that picture. Anyway, so they have tubules throughout their body. That actually pump water out. Okay, so they actually have holes in the side of their body, and these have these tubules that are constantly kind of taking the water and pumping it back out. And so it takes energy. So even planaria are osmoregulators. So instead of having all of the tubules in the kidney, they just have them throughout their body. And the same thing happens with earthworms. So each segment of an earthworm, it has its own tubule that pumps water out. Okay? So sometimes you see on rainy days, the earthworms coming to the surface. And why are they coming to the surface? They're going to drown, right? Because they actually breathe through their skin. So the earthworm's breathing through his skin, and then all of a sudden there's too much water in the soil, and there's not enough air, right? So the soil gets saturated. So the earthworms come up because they're drowning, right? They can't get enough oxygen. And so then you'll notice that they look bloated. And why do you think they look bloated? Excess water, right? So they've gotten too much water has moved in via osmosis, and maybe they're too stressed, or maybe they're dead, right? But they're not able to get rid of the extra water. So that's why you have these bloated earthworms laying out after a rainy day, right? Okay. So the same thing is that you have these tubules in every segment. Now we have actually created tubules and put it in an organ, and that is our kidney. Okay. So we can look at what happens in freshwater versus marine fish. So this is freshwater versus marine fish. Okay. So the same thing as the planaria and as the earthworm example, if I am in a freshwater environment, I am going to get water taken up through my skin, right? So if we look at freshwater, right, it passively gets water. Right? It drinks very little. It doesn't need to drink because it's just being absorbed across the surface of the gills and across the surface of the skin. Okay, so it passively gets water. It drinks very little, but it produces lots of dilute urine. Right? So here it's excreting lots of dilute urine. So it's getting rid of the water, the excess water, by urinating it out. It also has to pick up ions. So it has to pump sodium ions specifically. So it pumps, that takes energy, sodium ions in through the gills. Okay, so that's my freshwater fish. Okay. So if we look at the opposite, and if you think about it, salmon actually have to go from marine back to freshwater, and so their physiology has to completely reverse itself. So if we look at the marine fish, right, it loses water passively, so just through the surface of its skin. So it must drink lots of water. And then what it does is it gets rid of the salt by pumping it out. So really, it's a desalinization plant, right? So you're taking in, if I'm drinking salt water, I have to get rid of those extra sodium chloride, right? So the ions get pumped out. So sodium gets pumped out through gills. And I would conserve water by producing very concentrated urine. So conserve water by producing concentrated urine. So it's pretty amazing physiologically, anadromous fish that move from freshwater to marine and back again, because they have these completely opposite physiological processes that have to occur. Okay. So one of the interesting things are sharks. And I've never eaten shark, so I don't know, and I've never actually prepared it. But sharks, unlike marine bony fish, do not lose water. So they don't lose water. And the reason is that they retain urea, which is a solute, it's nitrogenous waste as well, in their body fluids or in their body tissues. And this makes them the same concentration. So they have the same ion concentration as the outside. So they do not lose water, right? So they are the same concentration as salt water. So same solute concentration, salt water. So they say that before you, if you're preparing shark meat, is you want to soak it in water to try to get rid of the urea. Because urea is a nitrogenous waste that probably wouldn't taste very good, right? So you want to get the urea out of the meat, right? Urea is toxic. And so the cool thing is the sharks have developed resistance to the toxicity of the urea. So it doesn't kill their cells. And they conserve energy. So this is a conservation of energy because they don't have to pump the ions back out. So this is a strategy to conserve energy. Osmoregulation in sharks and other cartilaginous fish like skates and rays and ratfish too. So it's just a different strategy than the bony fish have. Okay. So when we look at marine birds and reptiles, they take in lots of salty water. And if you think about it, they're eating lots of probably salty food. And so if they're marine, they have to have a mechanism to get rid of the extra salt. And so they have what are called salt glands. And these are designed to kind of clean their blood of salt ions. And so it is a special system that is set up as a counter current exchange system between the water and between the circulatory system. So you'll notice that blood is going in one direction, and then this water, this secretory tubule, it would be like a sweat gland, is going in the other direction. So notice that it's a counter current exchange. And so salt will just passively move from the blood into the water in the duct of the secretory vessel, and then it will be secreted. And so here, their glands sit up here, and then it gets secreted kind of out, and here in the seagull, there's a little channel in the beak that would then channel the salt water off, okay? So this removes salt from the blood. via a counter current exchange with watery secretions. So this is why sometimes it appears that specifically like turtles, marine turtles, look like they're crying, right? So if we look at a marine turtle and where its salt gland would be, okay. So this is a marine turtle, right? And so it's losing, this is its salt, this is where its salt secretions are. So what that is that's not, it's not a tear in the terms of the tears that we cry, but this is just a mechanism for them to get rid of excess salt, okay? So that's part of their salt glands. Okay, so on Wednesday, we'll start talking about the different types of nitrogenous wastes that are produced by different animals. So like the birds, for example, have uric acid, which they get rid of with their digestive waste, their feces, and that's why bird poop has white color in it because of the presence of its nitrogenous waste. And who wanted the TED Talk? I think I just printed it out. Let's see if it's still there.
Well, it almost got out. What? So fucking pissed right now. Report. Yeah, the midterm. But I'm keeping on the tool. But then, so he started laptop or laptop. And then went to work today. It's like, hey, so I got an email from so-and-so. And it says something about what this is. Sent an email about text to text. I sent it to him. Just a little bit. That's all I would do. So now I gotta wait till does my dad get home. But I have to report to that guy. Well, I see about it. That would shit happens, bro. Do you know the guy? No. That's all I would say. The email was close enough to his where it was. Yeah. How's it going?
All right. You?
That's so good.
How come?
I really typed your email address in wrong. Sent it to the wrong person.
Ah, you got the quiz to the right place, though.
Yeah. So I used our laptop instead of mine. So
Ah,
I got a bunch of them.
send it to me, Colton. Get it to me ASAP.
Yeah, I got it. I think my dad sent my head saying, did you send a paper to so and so because they don't know what this is for? Which means I just sent my other quiz to the wrong person too.
That sucks. I'm sorry. Bet she didn't mean to leave this up for me.